Good morning. Can you hear me? Yeah? Okay. So, welcome. Thanks for being here. In the next 1 hour, 1 hour and 30 minutes, we will go in a hands-on tutorial on brain disconnectivity. The first part, Dr. Salvalaggio will give a theoretical framework of what we mean for disconnectivity. Meanwhile, if you download the data, we save time for later. So if you have your laptop and you want to try all together, you can go on this QR code, download the Lesion. It's a Google Drive, and you can download the Lesion. It can take a while. And then we are going to use this toolbox. So download the toolbox, not this one. We don't have time for this. So deactivate. So QR code for the Lesion that we are going to use, and the toolbox. That is the BCB toolbox from Michel Thiebaud-Chotin. That is here. Hello, Michel. And just put in download. That takes time. Okay. Make a photo maybe that I will give the floor to Dr. Alessandro Salvalaggio.
Hello. Good morning. I am Alessandro Salvalaggio. I am a neurologist. So, first of all, welcome here in Padova. I hope you will enjoy the next few days here in Padua. I suggest you just to walk around in the city center. I think it is nice. You can taste the spritz. It is the local aperitif, the typical aperitif from Padova. Please don't ask me any information about the spritz. I'm an expert on spritz in Padua. And I will give the theoretical framework. So mine will be the boring part. Then Lorenzo will lead the hands-on session, so the funny part. Well, this is a very general definition of brain connectivity. It is the whole system of connection within the brain. We can look at brain connectivity from structural point of view. So the whole system of white matter tracks connecting different cortical areas that are measured with tractography, usually with DTI, diffusion tensor imaging. Or we can look to brain connectivity from a functional connectivity point of view, which is the temporal correlation of neuronal activity between brain regions, and this is usually measured with resting state fMRI. We can figure out the connectivity as the structural connectivity as the railways and the functional connectivity as the rail traffic. So we can easily understand that if we have a lesion, a problem, in any of the point of this connected system, we will have some problem also far beyond from the lesion site. And this is what happens in a patient when a lesion, a focal lesion occurs, for example, a stroke. If a stroke occurs somewhere in the system, the effect will spread far away, and also the cognitive function, the clinical outcome will correlate with the whole brain alteration caused by the focal lesion. But of course, the lesion itself, I miss the lesion, the topology. The lesion topology is important in order to explain the behavioral neurological impairment after a focal lesion, after a stroke, for instance. And there is a centuries and a half of study in this topic, the lesion symptom mapping and so on. You know, an example, you have some patient with the same clinical phenotype, with the same neurological deficit, and you compare the different lesions, you find where these different lesions overlap, and so you associate, you find that specific region, the overlapping region, is the region underlying the function that is impaired in that patient. Okay, is it clear? But sometimes it happens that you have some patients with the same clinical phenotype after a lesion, but the different lesions in a different patient are not in the same place. So you have the same clinical phenotype, but lesion located in different place. So what is going on? The explanation is this patient have in common the pattern of functional cognitive impairment, and also they have in common damage of the same white matter tracks. Usually, this happens for cognitive function. Okay. Well, our course in the last 2 or 3 decades, we improve a lot our knowledge on brain connectivity. Okay. But how is your feeling about the impact that this increased knowledge in brain connectivity have in clinical practice, in clinical setting? Your feeling is that brain connectivity is helping us in clinical setting. You look at, do you think that these 2 doctors are discussing about what is going on in brain connectivity or their patient? No, this is not happen. So this is some example of this guideline, American guideline for the stroke and the European guideline for neuro-oncology. And if you search in the PDF files how many times the term connectivity is mentioned, you will find that it is never mentioned. Okay, so structural and functional connectivity currently does not work in clinical practice. Okay, why? Oh, I don't know. I have some hypotheses. One hypothesis is that the acquisition of structural and functional connectivity requires a lot of time, because tractography and resting state fMRI requires a lot of time of acquisition, and in clinical setting we do not have time. Then it is difficult, it's hard to do the processing, okay, and the processing itself requires time, and in clinic we do not have time. And probably we have a lot of solid data at group level, but in clinical setting we need the solid data at the single patient's level. And then another possibility is that there is a cultural gap, a knowledge gap between clinician, neurologist, and between neuroscientists. Okay. Usually we use different languages and we have different aims. So this is a possible explanation why what we learn, what we study as neuroscientists is scarcely applied in clinical practice. So this is important. Event like this one, when we can talk together between clinicians and neuroscientists, are important to fill this gap. Well, so a possible solution was just to study the disconnection from focal lesion without using tractography and resting state fMRI. How is it possible? This is a method proposed by the group from Mike Fox, Harvard University, some years ago. It is based on ATLAS, normative ATLAS, in this case of resting state fMRI. They use hundreds of resting state fMRI from healthy subject. They perform an average ATLAS, an average map, in which they average for each single voxel of the brain the time course of the Bolt signal. And then from this atlas, they just overimpose, they just put on this map the lesion of the patient that they were studying at that time. And then they perform an average of the Bolt signal time course within the lesion ROI. And this is the point, the starting point, where they do the correlation from this ROI to with all the other voxels of the brain. So we explain this together because this is the key points for this indirect method. You use a normative data set. You can use any kind of normative data set with hundreds or thousands of healthy subjects with resting state fMRI. You perform an average of the Bolt time course for each voxel, and then you put the lesion of your patient, the normalized lesion of your patient in the MNI space, in this average map obtained from an atlas of healthy subject. And then you perform any kind of correlation from the ROI of the lesion to all the other voxels of the brain. And the interesting thing is that you just need the T1 weighted MRI, just the structural images from the patient. So each patient with a stroke will have a T1 scan, a structural scan. So potentially you can include every stroke patient from around the world. This is some example, just some example of the paper published by the group of Mike Fox with this approach. So, you know, I don't know if this approach will change our work in clinics, but of course this approach was successfully when we think about publishing. You know, this is some example. You find paper in JAMA Neurology, in Nature Medicine, and so on, and also a review in the New England Journal of Medicine. Okay, a couple of years later, the group of Michel Thibault Deschotten, that is here, developed a similar method, but with structural connectivity. So, same wise, you use a lesion of your patient, just the structural lesion of your patient, and you overlap this lesion in an atlas from healthy subject. In this case, the atlas is from tractography, so structural connectivity. In the original paper, it was with 10 healthy control. Now you can improve these atlases with any kind of healthy subject from any kind of atlas. So you can use hundreds or thousands of tractography. And you obtain these disconnectome maps. So you see in blue, this is the lesion. And in red yellow, there's a map of disconnection. So for each voxel, you have a probability from 0 to 1 of being disconnected. That voxel, that specific voxel was disconnected from the lesion. I suggest you to have a look, to give a look to the BCB Toolkit, BCB Lab website of Michel Thibault Deschotten. There are a lot of updates from the original method and other kind of indirect approach to the study of disconnection. Well, in 2020, these two approaches, the indirect functional connectivity and the indirect structural connectivity, were quite novel and promising for a translation clinical setting. So we decided to try to test them and to see if they were able to predict behavioral deficit after a focal lesion, after a stroke. So we aim to identify and quantify, given only a T1 lesion imaging, so the lesion that currently used in clinical practice, the most reliable approach among lesion, structural disconnection, and functional disconnection to predict behavioral impairment after stroke across multiple domains. Well, we use a dataset from St. Louis University, from the Corbetta group. We computed the structural disconnection map, the functional disconnection map. For the atlas of healthy subject, we use a 7 Tesla from the Human Cognitive Project with 176 subjects. And we test some behavioral score, motor right and left, visual loss right and left, language, memory, attention. So this is an overview of the method. We start from lesion maps, structural disconnection maps, functional disconnection maps. First we perform a principal component analysis in order to perform a data reduction. Then we test the prediction of the behavioral score with the ridge regression with Lee-Wanao cross-validation. And these are the two kinds of results that we obtain. This is a scatter plot with the measure of the spleen variance of the prediction of the behavioral score. And then these are the maps with the projected beta weights from the ridge regression. So these were the example of the result for the language score. You know that the prediction from the lesion and the structural disconnection were pretty good. A variance explained of 0.41, 0.48, while you can see here that the prediction from the functional disconnection was not good, 0.06. We performed the same analysis for the different behavioral score. And also in a subset of patients, we also compared the prediction from the direct resting state fMRI, because we also had the direct resting state fMRI. This is the overview of the results. You can see that lesion and structural disconnection were reliable in a good prediction of the score, while function disconnection predicted the behavior score the prediction was very, very low across the different domains. When we compare the prediction from the indirect function disconnection and the direct resting state fMRI, we saw that the prediction from the direct approach was much better than the one from the indirect approach. So there was some discussion about this method, but we can skip. So the result was that the indirect functional disconnection approach was not able to predict the deficit, the behavioral deficit post-stroke. Why? Well, a possible explanation is the low dimensionality of functional disconnection. Low dimensionality is it is because functional networks are biologically low dimensional, but also because when you perform, this is an issue of the method, when you perform the average of the signal within the lesion region, you probably perform a mixing of different networks. So that was, in our opinion, there was a problem in this approach. And then another problem is that, you know, most of the lesion localized, most of the stroke occur in the white matter or in the subcortical nuclei. And you know that functional connectivity is optimized for the cortex. So with Lorenzo Pini, we try to improve the indirect functional disconnection method in order to avoid this mixing of network within the lesion. And Lorenzo suggested to perform a PCA, a principal component analysis of the ball time course within the region of the lesion, in order to select just the first principal component and then to perform all the subsequent analysis only in this particular region. You can see here, a lesion contains different principal components of the ball signal. So we call this principal component function disconnection approach. You can see you improve the anatomical accuracy of this approach, but the prediction was still low. Nonetheless, the 100 method are very, very useful when you are facing with very rare syndrome, because you know there are syndromes that are very rare, so occurs in 100 of patients in 10 years all around the world, and you will lose a lot of patients if you aim to perform a tractography in all of these patients. So this is some example. We try to apply this approach to anosognosia for hemiplegia syndrome and also for anosognosia for visual loss. So patients are not aware that they have a bilateral visual loss. And we find that there is some specific pattern of disconnection for the two different kinds of anosognosia. But more interesting, we found some common pattern of disconnection. So it means that there is a combination of context-specific and supramodal mechanism in anosognosia. So this lack of awareness of a disease. We also, with Lorenzo, try to apply this approach to neurodegenerative disorder. You know, neurodegenerative disorder is not a focal lesion, but we still have a focal peak of atrophy. So we start from this focal peak of atrophy, and we were able to identify three different disconnection patterns of three different clinical variants of Alzheimer's disease. There are different other indirect strategies, indirect approach that were developed in the last year. This is one proposed from Joseph Griffiths. This is a way to a sort of quantification of the disconnection from each tract. So for example, you can say that the corticospinal tract is disconnected by a lesion. How much is it disconnected? 5%, 50%, 95%? It's different from a clinical point of view. And this is a tool that you can use to measure this information. Other disease, brain tumor. You know, brain tumors is a focal lesion, but the behavior of brain tumors is different than the one of stroke, because stroke you have a necrosis, neurons are dead inside the stroke. Tumor can disrupt the tissue, but can also dislocate the tissue. And we saw that the indirect approach, under stratoconnectivity approach, are not able to discriminate the dislocation that tumor causes to the brain tissue and the brain tract. This is an example of glioblastoma, a malignant brain tumor. I think you are not completely familiar, maybe you are not completely familiar with glioblastoma. This is an example of glioblastoma. You can see it's different from a stroke or another focal lesion. This is a contrast-enhanced rim. Inside there is a necrotic core, and you can see a surrounding edema and a mass effect. You know, these ventricles is smaller because of the mass effect of the glioblastoma. Glioblastoma is this malignant tumor with a very poor prognosis of 15 months. There are a lot of prognostic factors that are commonly used in clinical practice and in clinical trial, including age, performance status, extension or surgery resection, EDH1 mutation, GMT methylation, and so on. But as you can see, any of the prognostic factors take in account the impact that the brain tumor has on the organization of the brain itself, in particular brain connectivity, and the brain structural connectivity, white matter tract passing across or nearby the tumor. Okay. There is a link between brain pathology and structural connectivity for brain tumor, because malignant cell, usually their behavior is to climb along white matter tracts, and that's one mechanism, probably the main mechanism, how the malignant cell spread along the white matter tracts and in the brain. And this is a recent paper published in Brain last year, a year ago. And they found that if you perform a tractography, a DTI, a pre-surgical DTI, you will see that the larger is the impact of brain tumor in the tractography, the shorter is the overall survival. There is a strict link between brain connectivity, brain structural organization, and the survival. So with Lorenzo, Professor Corbetta, a couple of years ago, maybe 3 years ago, we started reasoning about the link between the structural organization of the brain and the impact that the glioblastoma has in this organization. And we know that the brain is not the same. There are regions where the density of fiber is low and other regions of high density of fiber. So there are regions that are in the periphery of the system that are less connected, and regions that are more connected. The same wise, glioblastoma can occur in regions with a higher density of fiber or lower density of fiber. So our hypothesis was that glioblastoma located in areas of high connectivity are associated with a worse prognosis, because the cells, malignant cells, have more possibility, more probability to find some white matter tract to spread. So this is, with Lorenzo, we found this index that we call tract density index. This is another indirect approach to a clinical problem. from a cognitive point of view. So we create, from a normative atlases of hundreds of tractography, or DTI tractography, we created these average maps of density of white matter tract with the help of Michel Thibault-Schotter. So the result was this map in which for each single voxel, we have a number that represents the average number of white matter tract passing through that voxel. And then you just put the glioblastoma segmentation in this MNI average maps of tract density, and we will have a number, a dimensional number, the tract density index, which represent that, which indicate that specific glioblastoma is located in a region of high fiber density or low fiber density. We perform a study, and will not go into detail, but with this tract density index, we perform a study in 2 independent cohorts and explore the relationship with tract density index and the overall survival of glioblastoma, and also the ability of a tract density index to predict the overall survival. We find a significant relationship, significant correlation, in which patients with a higher tract density index value show a lower overall survival. And this tract density index as prognostic factor was more reliable than all the other prognostic factors that are commonly used in clinical practice and clinical trial. So we demonstrate that the impact that the glioblastoma has on the structural organization of the brain is relevant for the outcome. There are some other analysis, model of prediction with a lot of tests in order to demonstrate our hypothesis that there is a significant relationship between glioblastoma impact on the structural organization and the overall survival. And the most important thing is that we obtain with the indirect approach. So you don't need to perform tractography in this patient. It is difficult to perform tractography in this patient because patients with glioblastoma are usually, the compliance is lower than the patient with stroke. So you can obtain this index. This is a pre-surgical index. You can imagine your patient will perform an MRI. It is routine, clinical routine, a pre-surgical MRI. Then you can obtain online, at the same time, you can automatize this process, the tract density index. So this is very helpful. This could be very helpful for the surgeon because he potentially can tailor the surgical strategy according to the tract density index. Yeah. So with Lorenzo, we ask ourselves if also an indirect functional disconnection approach will have some will be helpful in the prediction of the prognosis in this patient. And we tried the same approach for the functional disconnection approach that I show you some slide here for the stroke. We applied it to the glioblastoma, and I will skip. We performed some analysis, but we did not find any correlation. So the functional disconnection approach was not helpful in predicting overall survival in glioblastoma. So, as a summary, this is a sort of a summary of the outcome prediction with the indirect brain connectivity measure. You can see here, this is the structural disconnection, indirect approach, functional disconnection, indirect approach. You can see our structural disconnection is helpful in predicting outcome after a stroke, and also with the tract density index is helpful in predicting overall survival in glioblastoma. While structural disconnection approach are not working as predictor of outcome in stroke and also as predictor of overall survival in glioblastoma. So in conclusion, this finding suggests a close relationship between structural connectivity, white matter density, assesses with Indirect approaches, and the impact of focal lesion. And as this is the last slide, then Lorenzo will do the funny part of this session. The main advantages of this Indirect approach is that they are broadly applicable, and this is a possible option for clinical setting, a possible option to fill the gap between the study of connectivity and the implementation in the clinical daily life. Of course, this is not a complete substitute of direct resistive fMRI and tractography. And then we can say that indirect structural disconnection seems to perform better than indirect functional disconnection. Probably because they're monosynaptic versus polysynaptic mechanism. And thank you for listening. And I thank the people which I work every day, and also Michelle, Thibault Schotten, for the input. I was in the lab of Michelle 5-6 years ago, for when the BCB toolkit was still not published. So thank you again, Michelle. And if you have any question now, or maybe it's better after the hands-on session, as you prefer. Also question about Spritz or Padova.
So question for Alessandro, or we move directly to the hands-on session? We move, okay. Okay, so did you download the lesion? If you have the laptop, did you download the BCB toolkit? Okay. So basically, we have seen before, during Alessandro's presentation, this paper. This is the brain paper from Alessandro. And in the next 40 minutes, let's say, we will see how to compute these maps that were discussed just right now. So the first part, I will show you how to use the BCB toolkit to have the lesion from the lesion that already done and you have downloaded in your computer, to have these indirect structural maps. Yes? Yes, absolutely. So the QR code, the big one, not the small one, because we don't have time. If you're interested, maybe we can talk later. So the lesions are in the QR code, it's a Google Drive. I hope you have all the permission. I set all the permission to all of you. And download the toolbox is the, well, also if you go on Google and you write BCB toolkit, BCB lab, you go to the same page. I will show you maybe. Can I? Okay, if you click here, you go in the website that Alessandro showed you. And here there is the BCB toolkit, functionetome, disconnectome. We are going to use the first one, BCB toolkit. But of course, you could have fun also to look all the amazing toolkit from Michel's lab. Okay? So, the first part, we are going to use this toolkit for the structural one. Then, there are not available toolkit, or probably yes, but I don't know. For the functional one, we are going to do by, let's say, by hands with a little bit of coding, but it's very simple actually. Probably with the functionetome, you can do something similar. I don't know because I didn't check, but by the way, yes, true. Yeah, so you can do the same with the functionetome, but I will show you by hands, but more or less it's the same approach. Okay, so this is a Jupyter notebook. It's all Python, but you can reach the same results with R, C++, MATLAB, Julia, whatever, okay? Just one of your preference code. So basically here I'm going to upload some libraries that I use. Don't look at the red part because I have to upload everything, but I will do later, I don't know. Okay, so basically what we are going to see, it's if you download the lesion. Do you have the lesion? Do you have the lesion if you have the laptop? Okay. If you have some imaging toolbox to visualize, or you can download the MRI Cron. Maybe show you. So if you want something, can be this one. It's very simple. Now, since I don't know your level, and I guess it's a heterogeneous class, I will stay just above the surface. I'm not going to much details, but if you want something more, if you want some more tips, technical details, maybe we can talk later. So if you want something to visualize this kind of maps, which are the format is called Nifty, you can use a MRI Cron, or you can, if you have FSL, FSLIs, whatever. It's just a visualization tool, because basically what we have, it's, sorry. So basically lesion, when we have this lesion, what we have is numbers. So it's, what we have is this, it's an object, the lesion, it's an object, it's this Nifty, in which inside there are numbers. It's a matrix. In this case, the matrix is, the dimension is 182 voxel by 218 by 182. So when Alessandro before was speaking about, we take the lesion, we compute something inside the lesion, and we correlate with all the other part of the brain, we are talking about these numbers, okay? And basically, inside this matrix, there are numbers. The lesion numbers are just 1 and 0, because 0 means that the voxel is not lesioned, 1 means that you have the lesion inside that specific position in space, okay? And if I want to visually see the lesion, that's something that we have. So where is 1, I have a color, can be red, black, whatever. And if I have 0, I don't have anything. And if I overlay with a template, I can see the brain. Okay. So this is what a lesion, when you have an MRI, what you segmented the lesion looks like. And usually this is the typical image that clinician can observe. This lesion, of course, as Alessandro was talking before, must be in a normative space, because we know that each patient has different brains, bigger, smaller, different shapes, and we cannot use the lesion from the patient itself as the mask to compute this approach, but we have to normalize, to register, to make a deformation to match with a normative template. For sake of time, we don't have time to go into the details of this normalization approach, but you must be aware that the lesion must be in the same space of the connectome of the normative atlas that you are using. So if the normative atlas is in Hulick atlas, your lesion must be in the same space. If you use MNI, it must be in the same space. This is important, otherwise we are comparing lemon with apples, but we have to compare lemon with lemons. So the lesions that I provide are already normalized. So basically, I'll remove this. If I look at another patient, I have in the same space, now the lesion is in the brainstem. Okay, so the first example is a motor cortex lesion. The second is a brainstem. Then I don't remind. Sorry. Then we have a white matter, probably caudate lesion, you can see. And the last one is occipital lesion. Now these are two examples. It's not real lesion, but I try to comparing the real lesion that we have in the lab, I try to do something similar. Okay, so this is the starting point. We have lesion, we have our patients, we have lesions. These lesions are in MNI, in this case, so are in a normal space. So you can compare all our lemons, let's say, and we can compare this lesion with each other. Okay, this is the premises, very early premises. Now, what we have is the template. This is my template. We know that the brain, structurally, this is how it looks like. But as we have seen in Alessandro's presentation, the brain has some special features. And when we see white, which means white matter, It's not the same across all the brains, but if we want to look behind the scene, let's say, we will see that let's change colors, something like this. This, now you can see, it's the white matter template. Okay, so from here, if you follow Alessandro's presentation, we already see that if I have a lesion here, it's going to end one here, so maybe they are also near in space, but the way what is behind the scene will make the difference, because a lesion here is going to affect one tract that is different from a lesion here. Here we will intersecate the completely different tracks, and also what he was talking about, the tract density index. So this means that the yellow means high density fibers. So in these regions, which is corpus callosum, we have more fibers in other regions. So as we move in the periphery of the brain, so we have a change not only in the fiber density, but we have also change in the pathways that we are going to intersecate. Okay, this means axially and in all the other. I don't know what happened, so we can close. Okay, so this is the same visualization, but just flat. Okay, so here we have one lesion, and now what we have to do is to estimate, because this is an estimation. When we talk about indirect approach, we are talking of a proxy of something that we don't know in the brain of the patients, but we can estimate. We can say, okay, with a good probability, since this is a normative connectome and it's similar, it's an average of all of us, if one of us has a stroke, probably the tracts which is affected can be individualized, can be predicted by this average map. So what we are going to do now is, we have a lesion, and we have to identify which are the tracks which are likely disconnecting these patients. And now, as you can see, we are not talking about the tracks here. But what we want to do is to identify the full track that is passing through the lesion, that is encompassing with the lesion itself. So what we have to identify, if this is the tract, like example, we have to identify all of these tracts. And this means that the lesion locally is producing some distal effect. So what we have is not just a hole in the tract, but the full tract is disrupted. And this is the example Alessandro was making about the railways. If I have a problem in Paris, the trains are not traveling also in Bordeaux. So the problem is also in Bordeaux. And what we want to identify is the full path from Bordeaux to Paris, or probably Amsterdam. I come from Bassano de Grappa, probably you don't know where is Bassano de Grappa. If I have a problem in Bassano de Grappa, the only person that knows there is a problem is just me, because it's peripheral. And so probably the path goes from Bassano to Vicenza. But what we want to do is to identify exactly this path. Bassano, Vicenza in one patient, Amsterdam, Bordeaux in the other one. Okay, is it clear, this point? While when we are talking about the tract density index, that's a local measure, and it's the average of the fibers that are inside this mask. So it's not a path, but it's how important is the train station. How many paths you have in that specific train station, which is probably the railways in Paris, I don't know, 22, in Bassano de Grappa, just 2, okay? Now we are going to identify the tract that we have. So, let's see. Questions? This is very preliminary to understand the framework. Okay, basically what we have is this. Okay, so these are the paths of the brain, okay? These are our railway system. Now what we want to do is to identify in this chaos, in this chaotic path, which actually it's complex more than chaotic, if a lesion is going to overlap with one of these tracts. Now let's assume, these are just centroids, but let's assume that the lesion is here, okay? What I have disconnected, sorry. No, this is going to make a, wait. Sorry, the RAM of my computer is not very good, sorry. Okay, let's try again. Okay, so I have, so let's assume I take this track. So my lesion is here, okay? So what I have is disconnection of the full pathway, okay? Not just where is the mouse, but you can see that this connection is going to take several fibers, going from Paris to Bordeaux, okay? While, sorry. Okay, probably I have done an example here, which is better. Okay, let's do this. Sorry, but for this poor computer, there's no more RAM. Okay, so this is the example. So if I have the lesion here, okay, let's assume I have a lesion here, which is very focal. What I'm going to disconnect is the full corticospinal tract, okay? Now, the point is this, is that the corticospinal tract, my corticospinal tract, can be very similar to yours corticospinal tract, but there could be some heterogeneity, okay? So there are some points which, let's assume that the normative connectome is made by 10 subjects. If all the subjects in the space that we have seen, in this normative space which is done by this matrix, exactly at that coordinate, we all have a fiber passing in that exact voxel, and if we take a patient with a lesion in just that voxel is going to be disconnected by 100% of probability, which means that all of our normative subject has a disconnection in that exactly voxel, which for instance is in the cortical spinal tract is where we have the subcortical pathway. When we move to the cortical regions, there is more variability. So it means that maybe in a specific position in space, I have a tract, but maybe Alessandro, which is the other normative control, don't. So if we are just me and him at the normative connectome, and that patient has a lesion there, the probability of disconnection is 50%, because I'm the only contributor of this disconnectivity. So when we are going to talk, this is how Michel and the teams developed the toolbox, it's about the probability of disconnectivity, okay? So, yeah, and this is what I was talking about. Do you see the lesion? Okay, let's modify. Okay. So, yeah, now you see. So, the motor lesion that we have is in that position and is affecting the cortical spinal tract, right cortical spinal tract. So what you want to do is so this is just a visual example of what I was referring. So the lesion, it's in a position in the space, is in a train station, but not the left one, of course, but it's affected the full tracks that goes from the top to the bottom of the brain. So what we have to identify now is exactly that cortical pathway, okay? Question? Now, how to do? Well, thanks to Michel groups, it's very easy actually. What you have to do, did you download the toolbox? So, the toolbox is inside, I downloaded the Linux version, but there is also the Mac version, so you can use as you prefer. So inside the toolbox, there are several files. Let me show you. And there are some, the tracks that has been computed, for instance. So the decision that the toolbox is performing is based on a priori decision of the tracks. You see in green the tracks that we have. And here there are then the normative subjects. So just to run, well, for me it's a bash script, so I have to run as bash. But I guess in Mac there is an executable. I don't know because I don't use Mac, but you can try. By the way, this is the interface, okay? You can perform the normalization. So if you have your lesion in native space, you can perform the normalization. There are other functionalities that, for sake of time, we can see all of them. But what we are interested in is that disconnect on maps. So you just click in disconnect on maps. You have this GUI. And here it's very simple because you have just to point to the lesion directory. So for instance, I put the lesion in BigBrain, I guess, lesion folder. Let's check. No, it's not a directory. So you can navigate. Desktop, BigBrain, and lesion are my output, are my lesions, the 4 lesions that we have seen. And here you have to select a directory. You have to define the directory before and output. I save. And here we are. We have everything to perform the disconnectivity maps. Now there is a threshold, which is 0.5. This is the probability that you want from your maps. So 50%, at least in half of the sample, there is a tracks passing over there. We can apply the threshold later. It takes more time, but not too much. I mean, it depends how many lesions do you have. If you have thousands of lesions, you can go take a coffee, probably a lunch and dinner, and go back the day after. Here we have 4 lesions, so we can just run. You just run, and the magic happens. If you want to try. So. And you have an error. Now, there is an error because in the folder I have already computed yesterday, so don't worry, it works, okay? So what you have is output, okay? No, output, yes. Yes, because in the lesions, what I have? Yeah, because in the lesion I have something different, okay, but never mind. So, at the end, these are just something more that I put it. So you will have a logs file where you have all the processes that have been run, and then for each lesion that you have in the input, you will have the disconnection map, which is called in the same way. So for this is lesion 1, because I had a map in the lesion, you see, which is called lesion 1. Now the error occurs because of the space that we were discussing before. Because the BCBL toolkit has the MNI 1 millimeter resolution inside. So if your lesion is in MNI, but the space resolution is 2 millimeters, so it means that the box that we have seen is not 200, 182, 218, and 182, but is in 2 millimeters, that is 91, 109, 191. There is a spatial mismatch, and you have the error, okay? But why I have also lesion in 2 millimeters? Because for the function disconnection that we are going to apply, my normative is in 2 millimeters, because it takes more, I mean, it's more computationally demanding, so we reduce the space. So if you have an error, it's never machine's fault, or 99% is never machine fault, but it's our fault. So just be aware that most of the time the problems are linked with the space mismatch, okay? So for this, I have seen it, because he tried to perform the disconnectivity of all these. So maybe for the first one worked because of the space, it's okay, but the second one is the same lesion, 2 millimeters, you have a spatial mismatch and it doesn't work. But since I computed already, we can see the results. So, the prediction we have seen before. So based on this, okay, so the lesion, so maybe I can deactivate the left one. So my lesion is here, the cortical cortex. Here what I have is just an intersection with a fiber bundle from a normative control. So my prediction is that I should identify with the toolkit these tracks and maybe something more. Let's see the results. Yes, it worked. So basically what we have is that specific lesion that we see in the bottom is disconnecting the cortical spinal tract that we see here, that based on our prediction we could expect. But there is also disconnection of the corpus callosum. So the effect of the lesion is not just by one direction, but it's propagated also in other directions. So it means that voxels basically most of the subject for which the healthy connectome is built there is a tract that connect that specific voxel which are lesioned with that specific tract. So these are the maps in Salvalaggio's paper which are used then for the prediction. Now we can see, now do you remind that the second lesion was brainstem? Was a very small lesion here. Well, this very small lesion here actually, even not like the first subject, but is disconnecting as well as the corticospinal tract. So these two lesions basically are very at the extreme. One is the motor cortex, the other is the brainstem, but there is some commonalities between these regions, between these lesions, patients, which is the tracts that are connecting them. So when Alessandro was talking about if I have two patients with the same clinical phenotype, a lesion very close, we can find the substrate of that specific clinical phenotypes. Now we could assume that two specific patients with the same clinical phenotype, but two lesions apparently completely different in terms of lesion, volume, heterogeneity, now we could have some commonalities that could explain potential homogeneity in clinical phenotypes. Then we had the lesion in the caudate, and this is the disconnection, which is, you see that this is white, because it means there are some subjects in the healthy connectome in which there is a tract passing over there, but not the majority. So the probability is much lower compared where it's red or dark red. And the last one was occipital one. Here we go. And this is the disconnection of this particular region. Okay, so with two clicks, we can go from one lesion to this kind of maps that then you can use for region regression, for PCA, for machine learning, deep learning, whatever. Okay? Now, we can also convert, but just for visualization. Now we can compare, okay, sorry. I have to go back here. Now we can compare the lesions because the output of the BCB toolkit is in volume, so in the same space of the lesion size, of the lesion that you are including, so in Nifty. There are several tricks that you can perform to have a nice visualization with fiber bundles. So here what I have done is I took the maps, the volumetric maps in terms of probability. You can now decide to set a threshold. For instance, here I want to visualize just the tracks which are disconnected in more than 50% of my population. You can see, so this is the full threshold. You see, this is the full threshold from 0 to 1. We can identify just okay. We can identify just the tracks which are disconnected in more than 50% of the patients, and we can visualize as volume, as tracks. And so this now it's the results, which is similar to the disconnected tracks from the motor tracks that we have seen before. Plus we have the corpus callosum. And so in this way, in 1 minute, we go from one lesion to the disconnectome of that specific lesion. Okay? Questions? 1 minute of the computer. And 4 years, 5 years, no, 4 years of Michel's work. So thank you, Michel, because in 4 years we now have 1 minute, and we can have, yes, of course, yeah. Okay, so let's see. Okay, in the last 10 minutes, now what we can see, briefly, is the functional disconnectivity. So the functional disconnectivity, the concept behind it's quite similar. Okay, we have a normative map, but now the normative map is not a static picture, but it's a dynamic picture because the fMRI signal is a 4D volume, so we have signal on time. And now what we want to use, what we want to do, is to assess that specific lesion, which in me that I don't have a stroke has a specific fluctuation in time, to see all the other voxels which show the same fluctuation on time, to reconstruct the map. If you're familiar with fMRI, resting state fMRI, it's a CDROI analysis, okay, CDROI-based analysis. But usually, the CDROI-based analysis is done using ROI in the signal of the same person with the voxel of the same person. Now what we do is the signal is from the normative connectome, and the ROI is defined on the patient. So the patient will define the space. The normative connectome will define the time of the signal. So merging everything together will give us these functional disconnection maps. Before joining the Corbetto's lab, I was in the Alzheimer's disease field. And when I moved to stroke, then we talk about this indirect disconnection methodology. And at the beginning, for me, it was very difficult to understand. I mean, I use an ROI from one subject, and I use the signal from another subject. For me, it makes no sense at the time. But if you think, then I thought a little bit, when we do blood examination, we cannot compare our levels with prior a specific disease, but we have to compare our levels with a normative range. And so more or less is the application of this concept to the fMRI. So I use the CDROI from one patient, and I want to compare with the normative population. This helped me a lot to fix this incongruency that I had in my mind. So, how to do? Now, we have to upload these maps. The normative maps here, we don't have an average disconnectome, an average map from the population, okay? Because now here we are using, at every iteration, one healthy controls at time. So it means that if I have 100 healthy controls, I'm going to compute the functional disconnection map from all of these healthy controls. So I will have 100 disconnectivity maps. Then we can do in two ways. One is a computation of a statistics, which is quite similar to what we have seen, which is a t-score, because you want to see across these 100 controls, everything that is significantly different from zero, to say, okay, it's homogeneously activate or connected. Or another approach, maybe it's easier, is to make an average. So the average is not done before, but it's done after, okay? So you compute the disconnection, then you do the average, but we will see how to do. So basically here, compared to the maps that we have seen before, we have the time dimension, which is 300. So now the space is 191, 109, 91, which is the MNI 2 millimeters, which just for computational demanding, because I have to compute the disconnection voxel by voxel. So if I have to compute the dimension voxel by voxel, 182, 200, it takes the double. So usually you downsample, but also because the fMRI usually it's acquired with these 2 millimeters, just to don't interpolate and create a new signal that actually does not exist. So basically now we have the time, 300. So it means that we have acquired, I cut the data because we have to do today, but these data are from Human Connectome Project, 7 Teslas is 3,600, but my computer cannot manage 3,600 volumes, so I just had to cut the first 300 seconds. So now what we are going to do is this. So now we have the lesion, the motor cortex lesion that you can see here. No? Well, you can see the last one, this black spot. And now we have time. So before I just show one static picture, now we have more pictures because it's time zero. Time zero, the red and blue is the bold signal, the fluctuation. It looks horrible, but just because for visualization, and we should put thresholds. But this is the proper preprocessed raw signal. And then we have, after, I don't remind, 200 seconds. How much I have done? 200 seconds. And there are some differences. So basically, the bold signal inside the lesion fluctuates. And when I say fluctuates, I mean this. If we do what Alessandro was talking about, the average of the signal inside the lesion. So basically, in time, from 0 to 300, which are the seconds that we are registering, we have the fluctuation of the lesion. And this is just the average bold signal, this one, the average at each time point. And the result is this time series. So now we have the time series of that specific ROI computed not in the patient, but in a random healthy controls. Okay, this is just one random healthy controls. So for that specific healthy controls, that specific voxels, which are lesioned in a patient, show these fluctuations in time. I repeat, in that specific healthy controls. Now, what we are going to do is going to compute for each of the voxels outside the lesion how the voxels outside the lesion are connected with this time series. But I repeat, in a healthy control, not in the patients, because I don't have the fMRI from the patients, okay? So I use the space from the patients, the lesion, the voxels, and I computed my seed ROI analysis in the healthy controls, okay? So what I have is a kind of, okay, let's assume that this patient would never had a stroke. How the fMRI looks like? We assume that more or less should be like the normative connectome. But here there is a problem that Alessandro was pointing before, because if the structural disconnection is more similar between us, the functional maps are not so similar. And so this could be also another problem about the prediction of the behavior, because we know that the intervariability of that functional connectivity maps can explain our behavior. So this could be one of the reasons also that the behavioral prediction from the fMRI is not so good, because we are using data from, I repeat, unhealthy controls, okay? So, now, we need a little bit of, here is not 1 minute, because we have 300 volumes, so for each voxel we have to compute, so for each voxel we are going to compute this, and this is our reference, so for each voxel outside the black spot, we have to compute what is the correlation in time. So it means how the time series of voxel 1, voxel 2, voxel 5 until the end is similar to this time series. And we just perform a Pearson correlation, just simple Pearson correlation. So each voxel from these maps, so for 300 volumes, we have just one value, which is the R, the Pearson correlation. Then if you want to use Spearman correlation, mutual information, there have been several ways to compute. Let's say that the classical approach is Pearson correlation, which give us the, okay, and this is the connectivity maps. Maybe it's better to see like this. Okay, so since we are in a normative template, again, I can use a normative template, and now I can upload the connectivity maps that was computed. Okay, so now we have that specific motor ROI lesion that you can see where it's yellow. This is the projection at the surface level. Basically, the disconnection, this is of course one subject, so you have this effect, but later we are going to average. So this map is not only disconnected the motor cortex, but if you are familiar with resting state fMRI, you can see that this is seems the topology of the so-called dorsal attention network. So as we have seen for the structural disconnection, This lesion, that the lesion disconnected not only the full cortical tracts and also the corpus callosum, from a functional point of view, we have this nice disconnection of a network that seems the dorsal attention network. When we perform the study trying to improve the anatomy, is because, as Alessandro was saying, here what we are doing is the average of the signal in the full region, but this region can overlap a little bit with white matter and with gray matter. And the mixture between these two signals is not optimal, because we know that the fluctuation in the amplitude from the gray matter and the white matter is quite different. And so this, when we compute just in the gray matter, this map looks better. But this is the classical approach, not the, let's call the enhanced one. So basically this is a random guy without lesions, normal, young, because we use young, using the lesion of that patient as a CWI. So if we invert the concept, is that lesion in that specific patient, what is likely disconnecting? Probably in that patient, this network doesn't work very well, because we made a hole in a region of this network. It's like, this is my path from Milano to Amsterdam. I pass from, I don't know, Vienna. I remove Vienna. So the full path, the full network is not, probably is disconnected. And now we have another, this is a kind of a feature engineering, no? We have a lesion, and we can see the lesion in a different, from different point of view. And then we have these new maps, these new metrics, these new values that you can use for region progression, again, deep learning, whatever. Try to see whether you can improve the prediction just from the lesion itself, because we know that the brain, the voxel in the brain are different also in terms of this kind of features. Okay? And what we do now is to compute for all the subjects that we have. Here we have 5 subjects. I already done. And then we do the average. Here, for sake of time, I decide to perform the average. And basically, this is the average map. So now the results is not as in our papers, because I used just 5 subjects. I removed 80% of the signal. But then the concept is that these patients, on average, has the disconnection of this neural network, which seems the dorsal attention. Now, if you use the full-time series, because usually we use 173 subjects, so then the average, it's anatomical much better, but for sake of time, it's just a toy example. Okay? Questions? I think. Yeah. It's almost time to go lunch, but if you have questions for me or Alessandro, we will be more than happy to answer. And thank you for your attention, of course.
I have a question.
Yeah.
Have you ever been able to see the finger frontal articular vesicles using the function of
You mean a maps that looks like the FOF? No, but probably, well, there are some works assessing the networks in the white matter, but the point is that
Lorenzo,
when we pre-process the data,
I'm
we
sorry,
remove...
Lorenzo, could you please repeat the question, please?
What?
Could you please repeat the question? We could not hear
Yes,
that.
basically if we can see at functional level, yes, if we can see at functional level the inferior fasciculus, inferior frontal fasciculus, and potentially I never seen, potentially we could, but it depends by the way we process the data, because this data, to look at the gray matter, we remove the signal, the average signal from the white matter, and this is going to reduce the signal of the time course of the white matter itself. The cortical. No, the cortical connection, yes. The cortical connection. I'm not sure 90%, but I guess that in our patients I could have seen. But I have to check, Okay, maybe later I can check. For the structural, well, several studies use 50% of threshold. Then, depending on the application, you can decide to use a higher threshold or a lower threshold. It depends basically depends by the scientific questions. But with 0.5, you are more or less in line with several studies on this topic.
So to follow up on that question, are you giving guidelines with the BCD Lab tools for people to choose these thresholds?
Well, that could be nice, and maybe something that Michelle could work on it. Yeah, no, but I mean, I guess it's difficult to give a guidelines because it's really dependent by the application. Because if you want something very sparse, maybe you need to increase the threshold. If you want to be very focal, while if you want to have an idea about the probability, well, you need the probability value. So you need also voxel with a very low probability. You don't need threshold. So it depends really the application. Yes.
It also happens that the viewer asks to see the results with different threshold. So you need, I agree with Lorenzo, it depends on the scientific question, if you need more sensibility or more specificity.
Absolutely, but we also know that with these methods, they're very dependent on the choice of atlases or the way your networks are built, how you select regions. So how do you reconcile all these different approaches, all these different ways to look at the problem?
Well, for fMRI, for the functional disconnection, we don't use atlases, so that's purely data-driven, and so we don't have this issue.
Yes.
For the structural, that's purely atlas-driven. So I think that, I don't know if there are some studies comparing the maps based on different atlases, but if not, maybe that's something that it's mandatory, probably.
Because you already have a good framework to do that.
Yeah, maybe that's good. Yeah. So thanks for the tip. Yeah.
When you run the DTI or even for the tumor,
how did you get rid of the problem that gives the DTI, the water direction, once that you have the stroke immediately after, or in any case soon after, or eventually during and before neurosurgery? I don't know if I got
the track density. You are referring to track density index, or you are referring to the track density index, or to the DTI?
Just to
direct DTI. Okay.
The DTI. Yeah. How did you manage for that?
Sorry, can you please repeat the question?
Allora, when you have a stroke, when there is a stroke, fresh in quote, acute, or there is a tumor. That is growing, and you collect the data for the DTI, sometimes you get nothing on close to, because of course water starts going crazy according. But necessarily, sometimes in any case you try to solve this problem doing what?
Well, in this workshop we try to solve this problem using indirect approaches. So just not using DTI at all. We do not use DTI at all in this.
Yeah, but usually, yes, we don't. We do not. Yes, we just take the signal, the raw signal from the patient itself.
and then you use the atlas.
Well, it depends by the approach. You can use just a probabilistic tractography, which is a tractography which is built based on the data of the patients. So if there are in a tumors, there is a displacement of the tracks, but virtually.
Not something that you can directly measure.
No, from the data you can check, yes, you can infer the eventual displacement. But that's an open point, whether...
Well, there are an experience from neurosurgeons which have the neuron in their neuron navigator. Sometimes they have the map from the DTI. For example, they want to know if the corticospinal tract, when a tumor is close to the corticospinal tract or to the other important endocrine matter tract. They would like to know where the tracts are in order to preserve the tract, and they use the the indication from the DTI in the neuronavigator, but only something as they don't trust totally in the information from the DTI, because inside the surgery they sometimes found some differences because of the displacement, because of the edema. Edema is a problem in this measurement, because tumor have a lot of edema.